हेलो एवरीवन, आज का टॉपिक है ग्रिफिथ्स एक्सपेरिमेंट। बहुत ही इंपॉर्टेंट एक्सपेरिमेंट है। ग्रिफिथ वाज एन इंग्लिश फिजिशियन, ये आपको याद रखना है। और ये याद रखना है, ही वाज वर्किंग ऑन ए बैक्टीरिया नोन एज स्ट्रेप्टोकोकस pneumoniae, एंड दिस बैक्टीरिया causes pneumonia in humans and other animals. Now you should know that this bacteria has two important strains. One is called as the S strain and the other is called as the R strain. S का मतलब यहाँ पर है स्मूथ, क्योंकि जब भी हम S strain बैक्टीरिया को ग्रो करते हैं द कल्चर मीडियम दे फॉर्म स्मूथ कॉलोनीज एंड R strain बैक्टीरिया दे फॉर्म रफ कॉलोनीज व्हेन वी कल्चर देम इन द ग्रोथ मीडियम. Next thing is that S strain viruses, they are virulent, they cause the disease pneumonia. R strain bacteria, they are avirulent, they do not cause the disease. This is what you have to remember. Another important difference between S strain and R strain bacteria is that S strain bacteria, they have capsule, which is a polysaccharide coat present outside the cell wall. This capsule protects S strain bacteria from the attack of immune system within the animal body. Thus, they are able to cause infection. Whereas R strain bacteria, they lack capsule. They do not have capsule. Hence, they are easily attacked by the macrophages of the immune system and are destroyed whenever they enter into animal bodies. So they are not able to cause infection. They are not able to cause pneumonia. This is what is a really very important point. Now, let's talk about the experiment performed by Fred Griffith. First step. Now here what he did, he took Lyo S bacteria in the syringe and injected it into the Lyo mouse. Mouse suffered pneumonia and died after some time. And on doing autopsy, on doing analysis of the blood sample from the dead mouse, he found that S bacteria were present in its blood. And this is expected. You have injected S bacteria into its body and you recover S bacteria from its blood. Now, here in the next step, he took heat-killed type S bacteria into the syringe, injected it into the Lyo mouse, and Lyo mouse remained alive. It didn't die because we have already killed S bacteria. So it was not able to cause infection. Now we have living type R bacteria. They were taken into the syringe, injected into the Lyo mouse. Mouse didn't suffer pneumonia because R strain bacteria do not cause the disease. Now, this point is a very important point. Now, he took a mixture of heat-killed S-type bacteria and living R bacteria in the syringe, injected this mixture into the body of the Lyo mouse. And he was expecting that the mouse will remain alive, but mouse suffered pneumonia and mouse died. And when the blood analysis was done, S-type of bacteria were recovered from the mouse, from the dead mouse. So, where from these S bacteria came from? Because we injected Lyo R bacteria and from blood were obtained S bacteria. So after doing a lot of analysis, after doing a lot of thinking, a lot of research, he concluded that R type of bacteria were converted into S type of bacteria through a phenomenon called as transformation. And he called the substance we changed R type bacteria into S type bacteria a transforming principle. But he didn't know the nature of transforming principle. He didn't know whether the transforming principle was DNA, RNA, lipid, carbohydrates, protein. No, he didn't know anything. He said that something which he called as transforming principle has converted R type of bacteria into S type bacteria. This was Griffith's experiment. In the next lecture, we'll talk about Avery's experiment. Avery and his colleagues proved the nature of transforming principle. They showed us what this transforming principle was. So this is all for today. Thanks for watching.